everyone, and welcome back to Development, our last lecture series. All right, let's get started. So first of all, for anything to develop, we first have to have a fertilized egg. An oocyte is viable for about 12 to 24 hours after ovulation, and a sperm is viable for approximately 48 hours. Excuse me, you can see this blue bullet point. Some sperm can survive greater than 48 hours, but it's very, very, very few of them. So again, for fertilization to occur, coitus or intercourse has to occur no more than 3 days before ovulation, about 24 hours after ovulation. And fertilization is when the DNA from the sperm fuses with the DNA of an egg to form a zygote. That is technically fertilization. So it is not just enough for the sperm to come in contact with the cell membrane of the egg, but it's when the DNA actually fuses. That's our technical definition of fertilization. Well, and this is not as easy as that might sound. So even though there are hundreds of millions of sperm that are in each ejaculate, a lot don't ever make it to the egg. Some will leak out of the vagina. Some are destroyed by the acidic vaginal environment, and that's a lot, like millions will be destroyed by simply the acidic environment of the vagina. Again, millions will not make it through the cervix. Then when they get to the uterus, once those that do, that uterine cavity is huge for a tiny little cell like this, like the sperm cell. So they're either dispersed, they don't ever find the opening to the fallopian tubes, or they're destroyed by the female's immune system. So again, we're talking millions in attrition. A few hundreds to a few thousand will reach the actual uterine tubes. And that journey can take anywhere from 30 minutes to 2 hours, which is actually a lot faster than maybe I would think. Before a sperm cell can fertilize an egg or penetrate the cell membrane of an egg, it must undergo something called capacitation. So this is a physiological change that occurs in the sperm cell. Again, if it doesn't undergo capacitation, it won't be able to fertilize the cell. Basically, during capacitation, the acrosomal cap is altered. So remember, there's that acrosomal cap on the head of the sperm cell that contains digestive enzymes. It needs to be prepared to make those digestive enzymes available. Otherwise, it won't be able to digest through the corona radiata, the zona pellucida, making its way to the actual cell membrane of the egg cell. The tails will also beat faster and harder, again, to kind of help push it through all these different layers. So again, just as a reminder, so here's the cell membrane of the oocyte. There is this zona pellucida as well as this corona radiata. So here you'll see the sperm cell. Here's that acrosome in red in this image. Again, it has to be ready to be able to push its way through the corona radiata, and then as it hits that zona pellucida, which is basically a protein-like layer, it has to be able to digest that before it ever actually makes contact with the cell membrane of the oocyte. Okay, the sperm are attracted to the oocyte, the ovulated egg. And that's due to these chemicals. There's chemotaxis that's occurring. As they will navigate toward the chemical cue being given off by the corona radiata. And this is just what I mentioned to you. They have to basically get through this zona pellucida before they can ever actually touch and make contact with the cell membrane of the egg cell. So all that acrosomal reaction is that those enzymes are released, and they simply digest that layer. That's it. I don't know why I have 3 slides on that, when really one probably would have sufficed. Once they do make contact with the cell membrane, there are receptors that will help bind it to the cell membrane, and then a different protein that will help insert it into the membrane, which is what you're seeing in this image here. There are blocks to polyspermy, meaning there are blocks to multiple sperm trying to fertilize the same egg. So only one sperm is allowed to penetrate the oocyte. So a block to polyspermy, another way of saying that is the cell is ensuring monospermy, or the fertilization by one sperm cell. So anyway, there's two different blocks. One is a fast block, the other is a slow block. The fast one is membrane depolarization, and that prevents any other sperm from fusing with the cell membrane. And it sets in almost immediately. It lasts, you can see this, about a minute, during which time it causes the slow block to then initiate. Basically what happens here is that the receptors that were on the cell membrane are destroyed, and any sperm that may have come in right after that first one and that are attached to that receptor but haven't been brought into the cell yet are simply released. And then there are these mucopolysaccharides, this mucus-like coating that coats the egg and that the acrosomes cannot digest. So it becomes an impenetrable barrier to any late arriving sperm. So here we have that image we just saw, kind of zoom back out, and again here you can see several other sperm are close, but not as close as the very first one. So again, those are released. We have our fast block, slow block that occurs. The one that was able to get there first is brought inside the cell. The tail detaches from the head, and that pronucleus expands so that it will continue the cell division. And you'll notice that the nuclei come together to form remember it's half from mom, half from dad, so you have to have both halves in order to continue cleavage, and then you form the very first daughter cells. Okay, so that's what you're seeing here. Once again those nuclei come together, we have fertilization occurring. So that very first cell division, we end up getting into a 2-cell stage. Each one of those 2 cells will divide. You get 4 cells. Each one of those will divide. You'll get 8 cells, 16 cells, so on and so forth. So you get like a solid ball of cells called a morula. So that's what you're seeing here. It takes a few days. So you'll notice that it's about anywhere between 4 to 7 days that this dividing ball of cells, now hollow ball of cells called a blastocyst, is going to implant into the endometrial layer. Okay, so you can see that. And again, it's being moved down the fallopian tube toward the uterus via peristalsis, as well as there's cilia along the uterine tube that are also helping move things towards the uterus. Right? And if I think I already said this, but let me just make it again very clear. Fertilization occurs in the fallopian tubes. Okay. So before the blastocyst embeds into the uterine lining, you'll notice that here is a single line of outer cells. There's this inner cell mass, and then there's this cavity. So that's what you're seeing here. So this is a blastocyst. It's a fluid-filled hollow sphere composed of this outer trophoblast, inner cell mass. This will help form the placenta. This inner mass will become the embryo. Okay. During implantation, those trophoblast cells are going to adhere, or basically stick to, the endometrial cells. They will actually digest the endometrial cells until it is fully embedded in the endometrium. And so you can see in this image here how the developing embryo is continuing to grow as it's been embedded into that lining. And then this part, anything in purple here, are the common sites of implantation. So how do contraceptions work? Well, you can have vasectomies, having what's known as tubes being tied in the female, IUDs and birth control pills. So in a vasectomy, basically what that is the vas deferens is cut and it is tied off. Because it's very external, just underneath the skin and the scrotum, it's a tiny little incision. So it's a very quick procedure for males. Obviously, this means that there's still the same seminal fluid from the seminal vesicle, the prostate gland, the bulbourethral gland. It's simply that the semen will not contain sperm, right? So basically, the male would be infertile. This is easily reversed. So if the male chooses to later have children, this can easily be, the tubes can easily be brought back together again. In females, this is called a tubal ligation. And I guess I can make this image a bit bigger. I'm not sure why that is so small on here. It's kind of the same concept, but it's a much more invasive procedure, again, due to the internal nature of the female reproductive system. And you can see here there's variations on how tubal ligation can occur. You can either cut and cauterize, so apply heat to basically cause a break between the tubes. You can cut them and then tie them off, or you can simply band them together like this, and then sperm and the egg can't get to one another, right? You basically put a kink in the hose, if you will. It is more difficult to reverse a tubal ligation in the female than the male because, again, it's a more invasive procedure. An IUD, you'll notice there's two different types. There's either a hormonal device or a copper device. You can see it is inserted into the uterus, and you can kind of see it's not just blocking passageways into the fallopian tubes, but it also will cause usually thickening in the cervical mucus that makes it very difficult for the sperm to get through the cervix. And you can see some of these, like the hormonal one, will also suppress the ability of the sperm to not just get to the egg, but it also suppresses the ability to release an egg, so it suppresses ovulation as well. But both of those basically do the same thing. They're going to prevent the cervix from being penetrated by producing this thick mucus. I'll just also add that anything that's copper also acts as an antimicrobial. So if a copper IUD is inserted, it is also toxic to the sperm cells as well as the oocyte. So it will basically, it's toxic to them, so it will destroy those cells, preventing pregnancy. There are two main types of birth control pills. There's ones that most of them will contain both estrogen and progesterone. Some of them only contain progesterone. But basically, the main way that they work is they basically either stop or reduce ovulation. If there's no ovulation, there's nothing for the sperm to fertilize. They'll also thicken the mucus, making it difficult, again, for sperm to penetrate the cervix. They will also thin the lining of the uterus. So if, in fact, a sperm is past the mucus barrier of the cervix, it did ovulate, that a fertilized egg would have a very difficult time implanting. All right, so I will stop there for right now. If you have any questions, please let me know.